Okay, this should be a much shorter lecture on mutations. All right, so at this point, you should have seen the first two lectures on DNA replication. That's the first lecture. And then the second one is on transcription and translation. Um, you'll notice that there's a lot of vocabulary, steps, but you should also know that students tend to do really well on this section of exam 3. The section that students seem to struggle on the most is the genetics part. But when they get to the DNA section of the exam, they tend to do very well. So it is a very complicated set of processes: DNA replication, transcription, translation. There's a bunch of rules, steps, enzymes. But if you can keep track of all the enzymes, if you can keep track of all the rules, where these things are happening, how to copy DNA, how to transcribe it, how to read the RNA to create protein, you'll be fine. Okay, so what happens when things don't go right and we make mistakes when we copy the DNA? Well, we call that a mutation. So a mutation is defined as any change in the nucleotide sequence of DNA. And the changes can involve really, really big changes, like an entire chromosome change. What would be examples of that? Well, you remember in meiosis, homologous pairs coming together, right? Two homologous chromosomes forming a pair. And then they're supposed to cross over, right? Or recombine. And one of them gives up something but doesn't get anything in return. And that's called a deletion. And that's called a duplication. Well, that would be an example of large chromosomal changes. That would be a mutation. On the other hand, if you only change one nucleotide pair, that's also a mutation, and that's a very small change. Whereas a large chromosomal change is like a really, really big change. You tend to see the effects of that a whole lot more, because that's a big deal. So what would be an example of a single nucleotide change? Okay, I'll give you one DNA sample like it's A, T, C, T, A, G. And then I'll ask you, how do you know this is DNA? And you should know that it's because it contains T's. All right, if you were to copy that, it would be T, A, G, A, T, C, correct? That's copying it correctly. What if we try that again with a different cell? And we could do it again. T, A, C, A, T, C. I think you see the mistake, right? That would be a mutation. That would be a single nucleotide pair. That's a pair. And you made a mistake. Okay, so let's start with that. Let's focus on these mistakes because we've already covered the large chromosomal mistakes. We did that when we covered meiosis. We already did that. So for this lecture, let's just focus on single nucleotide mistakes. Okay. In a single nucleotide mistake, so let's maybe create an outline. If you make a single nucleotide mistake, you can do something called a a nucleotide substitution. You substituted one for another, like I showed you in the previous slide. And that can lead to three outcomes. Types of mutations. You can have a silent mutation, described here. You can have a missense mutation, described there. And lastly, you can have a nonsense mutation, down here. And we're going to go over each one of those. What is a silent mutation? How is that different from a missense mutation? What's a nonsense mutation? So these are three types of mutations associated with a single nucleotide mistake, where you substituted one nucleotide for another. Okay, let's take a look at them. Let's go back here. Yeah, you might remember that. I'm going to clean this up. You might remember that in this case, we were looking at RNA to read it and turn it into protein, right? We were reading the RNA instructions to create protein. Let's say that we had this DNA sequence. Let's say that it was A, C, T, A, G, A. And when we read that to create RNA, we would have U, G, oops, maybe I should change this to an A. U, G, U, U, C, U. So that's the RNA. And I know it's RNA because it's got all these U's on there. All right, so that's RNA. And let's read them three at a time. UGU would be where? U, G, U. And that's going to give me the amino acid cysteine. And then UCU, that gives me the amino acid serine, SER. All right, so that's if I have the correct DNA, let's say. But let's say that I have made a mistake on the DNA when I copied it. So the original DNA that I was copying must have been T, G, T, T, C, T. That's the original DNA. And when I copied it, I made this DNA. And when I transcribed it, I made this RNA. And when I read it, I read them three nucleotides at a time, and I came up with these amino acids. But let's go back to that original DNA, T, G, T, T, C, T. And I'm going to start reading it again. I have A, C, A, and I make a mistake. T, oops. Leave it at T, G, A. So I made a mistake here. And then I'm going to transcribe it into RNA. U, G, U, A, C, U. And then I break them up into groups of 3. And I read them again. So originally it's UGU, UGU. And sure enough, UGU still, and I get the correct amino acid, cysteine, that I'm supposed to put down. But then I made a mistake, and now it's ACU. And that gives me threonine. And see, that's a different amino acid. What kind of mistake, what kind of mutation is that of the three that we looked at earlier? All right, well, let's look at the definitions. In a silent mutation, there's no effect. Everything goes on right. You make the same exact protein because all the amino acids are correct. You made a mutation, but it didn't change the amino acids at all. Well, in our case, we did change the amino acids, right? We were supposed to have serine, and instead we have threonine. So that's a change in the amino acids for sure. So it's not a silent mutation. Let's look at the next one. What if you change the amino acids because the RNA code, the codons are different? Well, that's called a missense mutation, and that's what we created. We created a missense mutation. When you change the amino acid like we did, that's a missense mutation. All right, so we got this one out of the way. That's a missense mutation. You saw an example of that. Let's take a look at a silent mutation. Let's go back to the original thing we're supposed to have. We're supposed to have this. This is done correctly. So it's done correctly on the left. That's what we're supposed to have. Let's look at another mistake. So remember, let's say that we did the first one correct, UGU. And let's say that in this case, we've made a mistake in the DNA replication that gave us the following, okay? Let's say we get UCC. And remember, this is RNA because it's got a U, or it's got U's in it. So the original that we're supposed to have is UGU, UCU. Let's make those better looking U's maybe. We're supposed to have UGU and then UCU. We still have UGU, but now we have UCC. And that's definitely mutation because it's a change in the nucleotide sequence. So let's find UCC. It's right here. And it still gives us serine as an amino acid. So we get cysteine here like we're accustomed to seeing. And then we get SER here. So there's a mutation. The mutation is right here. That should be a U. We can make it louder. See, there's over here. It should be a U, but instead I put down a C. And yet, I still got the same amino acid. And the reason I got the same amino acid is because it looks like you can have changes in the triplet codon, specifically at the third nucleotide, and you'd still get the same amino acid. And if you scan around the table, you'll find that that happens a lot of times. You can change the third one, still get arginine. You can change the third one, still get proline, leucine, valine. In other words, there's redundancy, right, in our codons that give you still the same amino acid. And it happens in other cases to a smaller degree, like right here, right here. But if you notice, in every case, it's always the third nucleotide that you can change. If you change the first or the second, that's very likely going to give you a different amino acid. And now that's going to be a missense mutation. But if you get the same amino acid, That's called a silent mutation because it has no effect at all. You get the same amino acid anyway. So this mutation here is safe, right? You can mutate your DNA, but as long as you get the same amino acids, it's not going to have an effect at all on you. So that's the safest mutation. A missense mutation, though, it starts elevating the danger effect. Changing one amino acid could potentially change the way the protein folds, and it may not work the way it's supposed to. That would be like examples of sickle cell anemia. You survive it, you lead a mostly normal, healthy life. But under certain conditions like low oxygen, your hemoglobin, protein, changes its shape, which changes the shape of the red blood cell, and it could lead to complications. And we've already seen the symptoms of sickle cell anemia. Okay, so we've seen a missense mutation that's potentially dangerous. We've seen a silent mutation, which is not dangerous at all. What's a nonsense mutation? All right, let's go back to our table. And maybe in this case, we'll clean it up a little bit of all this stuff. Okay, so in this case, we're going to do another mutation. Maybe I'll do this one in blue. All right, so remember, we're supposed to have, in the correct sequence, we're supposed to have U, G, U, and U, C, U. And just to be clear, like this is just showing you two codons of a very long chain of codons. We're just showing you two of them. Okay. What if we changed something that was UCU, this one? That's the correct one. What if we changed it and now it says let me say U. Yeah, let's change this one. What if instead of it saying UGU, it said UGA? This one's still the same, UCU. What if we change this one? I'm going to make it red. And we changed it from U to A. All right, let's find it on the table. Let's find UGU first. UGU is over here, and it's going to give you cysteine. But we inadvertently made a mistake on, in this case, the third nucleotide. And normally, you can sort of overcome that. But in this case, it says UGA, and it leads to a stop, which means you don't put down any amino acid after the fact. You stop. You stop translation. So after this guy, we won't read any of the other amino acids that or codons that follow. There could be bunches of them, and we're not going to read them at all. And that means that the protein that was supposed to be very long with a lot of amino acids, it's now a lot shorter because you stopped early. You inadvertently put in a stop codon too soon. And so you don't build the protein all the way. It stops short. And that's called a nonsense mutation because you put a stop codon in there too early. All right, so if you had to rank which is the worst mutation to have, I think we would agree it's the nonsense mutation, right? Because now you don't build a protein right at all. It's short or shorter anyway, and it doesn't fold the same way. It probably doesn't work the same way. It probably doesn't work at all. The missense mutation would be the second worst. You only change one amino acid, but all the other amino acids are right. And the silent mutation is safe. Okay, so that's if you do what kind of mutation? If you do a single nucleotide change where you did a substitution, and the substitution has 1, 2, 3 different outcomes: silent, missense, nonsense. Let's look at another variation of changing a single nucleotide, but we're not going to do a substitution. In this case, we're going to insert or we're going to delete a nucleotide. We're going to add a nucleotide that shouldn't be there, or we're going to delete one that's supposed to be there. And that's going to lead to something called a frame shift mutation. And what that does is it changes the reading frame. That's why we call it a frame shift mutation. The original codons are all going to change. You're going to need a visual for this one. Okay, this is the normal gene up above. Look at the RNA and then look at the amino acids in boxes down below. The amino acids form a protein. There's your methionine that starts it always. And then followed by 1, 2, 3, 4 more amino acids for a total of 5. Now look at a base substitution down below. We changed this G and replaced it with an A, and that gave us a different amino acid. I think you should recognize this as what kind of mutation if you substitute one nucleotide for another and it changes only one amino acid. Missense. But now look down below. What happens if you take out one of these uracils? See, it's being removed. That's a base deletion. And then look at what happens to the reading frame. So here's your first codon. That's still the same as what it's supposed to be. Here's your second codon. That's still the same as what it's supposed to be. But then what's the third codon look like? UUG, but it's supposed to be UUU. And then look at the next one. It's supposed to be GGC, and instead it's GCG, because everything has shifted over. And because you shifted everything over, every amino acid is different. So you frame shifted and created a totally different protein, not the one you intended. And who knows if it'll work. It very likely will not work the way you wanted it to work to begin with. You'll get some other protein, and it won't work. That's called a frame shift mutation. And we gave you an example of a deletion, but it can also happen if you insert something that shouldn't be there. It'll also shift the reading frame. It'll also change every single amino acid. Significant changes in the amino acid sequence, producing a non-functional polypeptide or protein. Okay, hope that made sense. So what causes these mutations? Well, the formation of a mutation is called mutagenesis, as in mutation generator or origin of mutation. And those mutations can be caused by what we call mutagens. A mutagen is anything that generates that mutation. And some of these we already know about, right? Like UV. Changes our DNA in our cells, causes potential tumors, cancers. X-rays can do that too, but We do a very short burst of an X-ray. We cover the body parts that are vulnerable, right? If you ever had an X-ray, you might have worn some sort of protective shield around your organs and stuff like that. And it's very quick, and it's not something you experience throughout a lifetime. It's only when you potentially broke something or we need to see a image of some part of your body. So even though X-rays are considered mutagens, they're quick burst, one time, and then that should be that. So we minimize significantly the potential effect. The most common mutagens would be chemicals in our environment, right? Like the chemicals in cigarettes, chemicals maybe in certain foods, other products. If you do some digging about carcinogens in your home, you'd be surprised to find that they're everywhere. In your carpet, your linens, detergents, carcinogens are everywhere. But the carcinogen is only an issue with prolonged high exposures of them. Some carcinogens are worse than others. So this is a big conversation, right? It's hard to just summarize it in a couple of lines. So if you're interested in carcinogens, you can do some research there. Okay, so we've covered mutations. What kind of mutation do you see here? The original DNA was CTT. We made a mistake, call it CAT. That changed the RNA codon from GAA to GUA. And instead of getting glutamic acid, now we got valine. Is this a silent mutation? Is this a missense mutation? Is this a nonsense mutation? Is this a frame shift mutation? What is it? You should have said it's a missense mutation. Okay, that completes the unit on DNA lectures. This was lecture number 3.